Right now, invisible particles are tearing through your body at nearly the speed of light. On Earth, our atmosphere and magnetic field stop most of them. In deep space, nothing does. Every second beyond Earth's protection, charged particles from exploded stars and violent solar eruptions pierce through spacecraft walls and slice through your DNA like bullets through paper. NASA discovered that astronauts on the International Space Station absorb radiation equivalent to roughly 10 chest X-rays every single day. And the station still sits within Earth's protective magnetic embrace. Venture beyond this shield into true deep space, and the exposure increases several-fold. Scientists have now calculated the exact timeline of human destruction from cumulative radiation, and the numbers reveal a terrifying truth about our cosmic limitations. If you are ready to discover why the universe itself forbids human expansion, hit that like button and subscribe. Now, settle in. Get yourself comfortable. Let's begin. Earth wraps itself in an invisible shield that most people never think about. This magnetic cocoon extends roughly 65,000 kilometers into space, deflecting the majority of charged particles streaming from the sun and beyond. The atmosphere adds another layer of protection, absorbing and scattering radiation that penetrates the magnetic field. Together, these barriers reduce your annual radiation exposure to roughly 3 millisieverts. That number represents your baseline, the natural background radiation you absorb simply by existing on this planet. 3 millisieverts per year. Remember that figure. It will become increasingly horrifying as we venture outward. The magnetosphere operates like an enormous invisible funnel, channeling dangerous particles toward the polar regions where they create the auroras. Without this deflection system, the surface of Earth would be bathed in radiation levels incompatible with complex life. The magnetic field has protected our planet for billions of years, allowing evolution to produce organisms that never needed to develop resistance to cosmic radiation. This is the environment that shaped human biology. Our cells, our DNA repair mechanisms, our cancer suppression systems, all evolved under the assumption of minimal radiation exposure. We are creatures of a sheltered world. Biological machines optimized for conditions that exist almost nowhere else in the known universe. Now, consider what happens when you leave. The International Space Station orbits at approximately 400 kilometers altitude, still well within Earth's magnetic embrace. The station circles the planet every 90 minutes, passing through regions where the magnetic field offers more or less protection. Astronauts there experience radiation levels roughly 50 to 70 times higher than people on the ground. They accumulate between 150 and 200 millisieverts per year. This elevated exposure occurs despite the station remaining firmly inside Earth's protective magnetic envelope. The magnetosphere is not a perfect shield. High-energy particles still penetrate. The South Atlantic anomaly, a region where the magnetic field dips closer to Earth's surface, exposes station crews to elevated radiation during each orbital pass. Astronauts report seeing flashes of light when they close their eyes in this region, caused by cosmic ray particles striking their retinas and triggering false signals in their optic nerves. Even in low Earth orbit, protected by the magnetosphere, astronauts accumulate measurable biological damage. Studies of long-duration station crews show increased chromosome aberrations, elevated biomarkers of oxidative stress, and changes in gene expression patterns associated with radiation exposure. These effects are detectable after missions lasting only 6 months. But even this dramatic increase represents a gentle introduction to what awaits beyond. Step outside the magnetosphere, and everything changes. Deep space begins, where Earth's magnetic field fades into irrelevance, roughly beyond the orbit of the Moon. The transition is not sharp. The magnetosphere has a complex shape, compressed on the sunward side and stretched into a long tail on the night side. but somewhere between 50,000 and 65,000 kilometers from Earth, depending on solar wind conditions, the protective bubble ends. Beyond this boundary lies the true radiation environment of space. It is nothing like what humans evolved to tolerate. It is nothing like what our bodies can withstand indefinitely. It is an invisible ocean of energetic particles that fills the entire solar system and extends throughout the galaxy. In this realm, two types of radiation dominate the environment. Understanding them reveals why cumulative exposure, not any single event, determines how long humans can survive. Galactic cosmic rays represent the first and more insidious threat. These particles originate from supernovae and other violent cosmic events occurring throughout the Milky Way and beyond. When massive stars explode at the end of their lives, they accelerate atomic nuclei to tremendous velocities. These particles then wander through interstellar space for millions of years, bouncing off magnetic field irregularities, gradually losing energy but never stopping entirely. The galactic cosmic ray flux at Earth's orbit consists primarily of protons, roughly 87% by number. Helium nuclei make up another 12%, but the remaining 1% deserves special attention. These are heavy ions, the nuclei of elements like carbon, oxygen, silicon, and iron, stripped of their electrons and accelerated to significant fractions of the speed of light. Heavy ions carry enormous kinetic energy. A single iron nucleus in the galactic cosmic ray flux can carry as much energy as a fast-pitched baseball, concentrated into a particle smaller than an atom. When such a particle strikes biological tissue, it does not simply nudge molecules aside. It tears through them, leaving a trail of destruction that extends far beyond its physical path. The flux of galactic cosmic rays remains relatively constant over human timescales. You cannot hide from them. You cannot wait them out. Every second you spend in deep space, these particles pass through your body in predictable numbers. Scientists measure this flux carefully using instruments on spacecraft, high-altitude balloons, and space station experiments. In interplanetary space, galactic cosmic rays deliver approximately 400 to 900 millisieverts per year depending on solar activity. The Sun's magnetic field provides some modulation. During solar maximum, when the Sun is most active, its expanded magnetic field deflects more galactic cosmic rays, reducing the flux at Earth's orbit. During solar minimum, when solar activity wanes, galactic cosmic ray intensity increases. This solar modulation offers a cruel choice for mission planners. Launch during solar maximum, and you face reduced galactic cosmic ray exposure, but increased risk from solar particle events. Launch during solar minimum, and you face the opposite trade-off. There is no safe window. There is only a choice between different types of danger, but these numbers describe average conditions. They do not capture the true horror of what these particles do when they strike biological tissue. That comes later. For now, understand that galactic cosmic rays deliver a baseline radiation dose that accumulates continuously, day after day, month after month, with no possibility of escape. The word accumulates deserves emphasis. Radiation dose is not like a sunburn that heals over time. It is not like fatigue that rest can cure. Each millisievert represents permanent damage to cells and DNA. Some of that damage gets repaired. Much of it does not. The body keeps a running tally whether you want it to or not. Solar particle events represent the second major radiation source. These eruptions occur when the Sun releases massive bursts of protons and other charged particles during solar flares and coronal mass ejections. Unlike galactic cosmic rays, solar particle events are episodic and unpredictable. Some years pass with minimal activity. Other years bring dozens of significant events. The Sun operates on an approximately 11-year activity cycle. During solar maximum, sunspots proliferate, magnetic field lines become tangled and stressed, and explosive releases become more frequent. A major coronal mass ejection can hurl billions of tons of charged particles into space at speeds exceeding 2,000 kilometers per second. When these particles reach Earth's orbit, typically 1 to 3 days after the eruption, they wash over anything in their path. Earth's magnetosphere deflects most of them toward the poles. Spacecraft in low Earth orbit receive elevated but survivable doses, but anything beyond the magnetosphere takes the full impact. During a major solar particle event, radiation levels in unshielded deep space can spike to lethal doses within hours. The Halloween storms of 2003 would have delivered approximately 2,000 millisieverts to an unprotected astronaut in interplanetary space, enough to cause acute radiation syndrome. Symptoms would include nausea, vomiting, fatigue, and declining blood cell counts. Recovery would be possible, but uncertain. The Carrington event of 1859 remains the most powerful solar storm ever recorded. Telegraph systems failed worldwide. Operators received electrical shocks. Auroras appeared as far south as the Caribbean. Scientists estimate it would have delivered roughly 10,000 millisieverts to anyone caught outside Earth's protection, a dose fatal within days. Death would come from catastrophic damage to bone marrow and the gastrointestinal tract, the body's most radiation-sensitive tissues. These extreme events capture public attention because they threaten immediate death. Mission planners designed storm shelters and warning systems specifically to address solar particle events. The warning time between detection and particle arrival ranges from 15 minutes to several hours depending on particle velocity. With adequate shielding and sufficient warning time, astronauts can survive individual storms. This leads many people to assume that solar radiation represents the primary barrier to deep space exploration. Solve the storm shelter problem, they think, and humans can venture anywhere. They are wrong. Solar particle events, despite their dramatic danger, represent an engineering problem with engineering solutions. Build a small, heavily shielded compartment. Monitor solar activity. Retreat to shelter when warnings arrive. The problem is difficult but solvable. Crews can survive individual events and continue their missions. Galactic cosmic rays represent a physics problem with no current solution. They arrive continuously from all directions. They pass through virtually any practical shielding thickness. There is no shelter that works. There is no warning that matters. There is only the patient accumulation of dose, hour after hour, day after day, month after month, and the cumulative dose from galactic cosmic rays is what ultimately determines how long any human can survive beyond Earth. NASA established career radiation exposure limits decades ago based on calculations of cancer risk. These limits emerged from studies of atomic bomb survivors, nuclear workers, and medical radiation patients. They represent the agency's best estimate of how much radiation exposure increases lifetime cancer mortality to unacceptable levels. The limits vary by age and sex, reflecting differences in radiation sensitivity and remaining lifespan. Younger astronauts face lower limits because they have more years during which radiation-induced cancers could develop. Female astronauts face lower limits because breast and ovarian tissues show elevated radiosensitivity. But all the limits cluster around 1,000 millisieverts over an entire career. Exceed this threshold, and your probability of developing radiation-induced cancer rises above what NASA considers acceptable risk. The current standard defines acceptable risk as a 3% increase in lifetime cancer mortality. This means that an astronaut who reaches their career limit faces 3 additional chances in 100 of dying from cancer, compared to an otherwise identical person who never flew in space. The agency has debated raising these limits, acknowledging that deep space exploration requires accepting higher risks. Some proposals would increase the acceptable risk to 5%. Others would abandon fixed limits entirely in favor of informed consent, letting astronauts accept whatever risks they choose. But biology does not negotiate. Raising administrative limits does not change what radiation does to human tissue. An astronaut who accepts twice the current limit does not become twice as radiation resistant. They simply agree to accept twice the consequences. Consider what these numbers mean for deep space missions. A journey to Mars takes approximately 9 months each way under current propulsion technologies. The precise transit time depends on launch window selection, trajectory optimization, and propulsion system performance. But 7 to 9 months represents a reasonable planning figure for chemical propulsion. Astronauts would spend roughly 540 days in transit, plus time on the Martian surface. During the transit phases, they would be fully exposed to the interplanetary radiation environment. Galactic cosmic rays alone would deliver between 300 to 900 millisieverts during the 18-month journey, depending on solar cycle timing and shielding effectiveness. This estimate comes from measurements made by the Radiation Assessment Detector aboard the Curiosity rover during its transit to Mars in 2011 and 2012. Add the time on Mars, where the thin atmosphere provides minimal protection and cumulative exposure approaches or exceeds NASA's career limits from a single mission. The Martian atmosphere is less than 1% as dense as Earth's. It provides roughly 1/4 the shielding effectiveness of Earth's atmosphere. Surface radiation doses average 230 to 270 millisieverts per year. Factor in solar particle events, which would likely occur during such a lengthy mission, and the numbers climb further. A crew departing during solar minimum would face elevated galactic cosmic ray flux. A crew departing during solar maximum would face frequent solar particle events. Neither option offers safety. Both options lead toward the same career limit threshold. The first humans to walk on Mars will almost certainly exhaust their career radiation allowance in a single trip. They will have no margin for future space travel. Their bodies will carry permanent damage that increases cancer risk for the rest of their lives. They will return to Earth as radiation casualties who won a race against a biological clock, and Mars represents the easiest deep space destination. Missions to the asteroid belt, to Jupiter's moons, to the outer solar system would require years of transit time. The main asteroid belt lies between the orbits of Mars and Jupiter, roughly 2 to 4 times Earth's distance from the Sun. A round-trip mission to a main belt asteroid would require 2 to 3 years of transit time using chemical propulsion. Jupiter orbits at 5 times Earth's distance from the Sun. A mission to Europa or Ganymede would require 5 to 7 years of round-trip transit. The radiation environment near Jupiter adds further complications. Jupiter possesses the most powerful magnetic field in the solar system, trapping charged particles in intense radiation belts that would kill unprotected astronauts within minutes. Each additional month in deep space adds more radiation dose to the running total. The mathematics is simple and unforgiving. More time in space means more radiation absorbed. Higher dose rates mean faster accumulation. You can reduce one factor or the other, but you cannot eliminate either entirely. Every improvement in one variable merely shifts the constraint to another. Double your shielding effectiveness, and you halve the dose rate, but you double the mass you must launch from Earth. Halve the transit time, and you reduce total exposure, but you must develop propulsion systems far beyond current capabilities. The fundamental trade-off remains regardless of engineering cleverness. This brings us to the real limit that governs human survival in deep space. Every proposed mission to every proposed destination faces the same fundamental constraint. Not the challenges of closed-loop life support or the difficulties of long-duration isolation. Engineers can design better recycling systems. Psychologists can select more resilient crews. Mission planners can provision adequate supplies. Radiation dose accumulates regardless of spacecraft design, propulsion technology, or crew selection. It accumulates whether astronauts are awake or asleep, working or resting, inside the spacecraft or outside. It accumulates from the moment they leave Earth's protection until the moment they return. The question is not whether cumulative radiation will harm astronauts. That outcome is guaranteed by physics. The question is how much harm they will accept and for how long they can survive it. Some readers might wonder about the Apollo missions. 12 humans walked on the moon between 1969 and 1972. They spent up to 3 days in deep space beyond Earth's magnetic protection. Why did they survive? The answer lies in timing and luck. Apollo missions lasted roughly 8 to 12 days total, with only a fraction of that time spent beyond Earth's magnetosphere. The lunar transit took approximately 3 days each way. Surface stays ranged from 21 hours for Apollo 11 to 74 hours for Apollo 17. The cumulative radiation dose from such brief excursions remained well below dangerous levels. Apollo astronauts received between 2 and 11 millisieverts per mission, comparable to a few CT scans. This exposure was measurable but not harmful. It fell well within background variation for occupational radiation workers. The brevity of the missions kept cumulative dose in the safe range. More importantly, no major solar particle events occurred during lunar surface operations. Apollo 12 launched within hours of a significant solar flare, but the crew had not yet reached the moon when the particles arrived. Apollo 16 crews faced elevated radiation from a solar event during their return journey, but they were already heading home when exposure peaked. No crew was caught on the lunar surface during a major storm. Had the August 1972 event occurred 4 months earlier, during the Apollo 16 mission, astronauts on the moon would have received potentially fatal radiation doses with no possibility of shelter. The lunar module provided minimal shielding. The spacesuits provided essentially none. Crews on the surface during a major event would have absorbed enough radiation to cause acute radiation syndrome. NASA understood this risk. Internal documents from the era revealed genuine concern about solar particle events and serious discussions about mission abort criteria. They accepted the risk because the missions were short and the probability of a major event during any given mission remained low. Probability mathematics works differently over longer durations. A 5% chance of disaster per month becomes a 46% chance over 12 months. Extend that to multi-year missions and catastrophic solar events become near certainties. The odds that protected Apollo become the odds that condemn deep space travelers. The Apollo astronauts beat the odds. They flew during a period of moderate solar activity. They completed their missions before any major events occurred. They returned home with stories of adventure and minimal radiation exposure. Future deep space travelers will face those same odds again and again, month after month, until the odds catch up with them. Yet even this focus on acute events misses the deeper problem. Solar particle events threaten death from massive acute exposure. A single storm can deliver lethal doses within hours, but storms can be predicted, at least partially. Shelters can be built. Operational procedures can minimize exposure during events. Galactic cosmic rays threaten death from cumulative chronic exposure. There is no storm to predict. There is no shelter to retreat to. The particles arrive continuously, 24 hours a day, 7 days a week, every week of every month of every year, spent in deep space. Both pathways lead to the same destination. Both add to the same running total, but cumulative exposure is the more fundamental barrier because it cannot be avoided or mitigated through operational procedures. Storm shelters can protect against solar particle events if warning systems work and astronauts can reach shelter in time. A heavily shielded compartment in the center of the spacecraft, surrounded by equipment, supplies, and water tanks, can reduce storm exposure by factors of 10 or more. Crews can survive individual events and continue their missions. There is no equivalent shelter against galactic cosmic rays. No practical shielding thickness stops enough particles to reduce dose rates to safe levels. Galactic cosmic rays pass through spacecraft walls, through equipment, through human bodies, and continue into space. They deliver their dose continuously, regardless of what astronauts do or where they hide. and their effects accumulate invisibly until the damage becomes undeniable. This is the invisible storm that bathes all of deep space. It cannot be seen or felt. Astronauts do not experience radiation exposure the way they experience acceleration or temperature. There is no sensation associated with a heavy ion nucleus passing through your brain. There is no warning when a galactic cosmic ray severs a DNA strand in your bone marrow. The damage announces itself only through biological consequences that take months or years to manifest. Chromosome aberrations increase gradually. Cellular repair mechanisms become overwhelmed gradually. Cancer risks rise gradually. Cognitive function declines gradually. By the time symptoms appear, the harm is already done. The dose is already accumulated. The future is already determined. Every millisievert absorbed in deep space contributes to a permanent biological record. That record cannot be erased. It cannot be reset. It can only grow larger with continued exposure. The body does not heal radiation damage the way it heals cuts or bruises. It merely accumulates the insults and waits for consequences to emerge. Understanding why this is true requires looking much more closely at what radiation actually does when it enters the human body. The mechanisms of destruction operate at scales far too small to observe directly, but far too significant to ignore. Individual particles trace paths through tissue that determine cellular fates. Individual DNA breaks trigger cascades that determine organ function. Individual mutations determine whether cells live or die or transform into something worse. The next section will examine these mechanisms in detail. But the fundamental point is already clear. Cumulative radiation dose represents the governing constraint on human survival in deep space. Everything else, the engineering challenges, the physiological adaptations, the psychological resilience, operates within the boundaries set by radiation exposure. Solve every other problem perfectly, and radiation still sets the limit. The invisible storm waits for anyone who ventures beyond Earth's protective embrace. It waits patiently because it has all the time in the universe. It waits silently because particles make no sound. It waits inevitably because there is nowhere in deep space that it does not reach. The question is not whether humans can survive this storm. The question is for how long, and answering that question requires understanding exactly what happens when cosmic radiation meets human biology. A galactic cosmic ray enters your body at roughly 90% the speed of light. In the time it takes you to blink, that particle has already passed completely through you and continued into space. The transit takes less than a nanosecond. A duration so brief that no human sense could possibly register it. But in that instant, the particle leaves behind a trail of devastation at the molecular level that can persist for decades. The particle itself feels nothing. It is an atomic nucleus, a collection of protons and neutrons bound together by forces incomprehensibly stronger than anything holding your body together. Your flesh presents no meaningful resistance. The particle passes through skin, muscle, bone, and organ as easily as a bullet passes through fog. It exits your body having lost only a tiny fraction of its energy, continuing onward into space where it may travel for millions of additional years before encountering another obstacle. But the energy it deposited during that nanosecond transit does not disappear. It spreads outward from the particle's path, breaking molecular bonds, stripping electrons from atoms, creating a microscopic cylinder of destruction that extends from entry wound to exit wound. Every molecule along that path is affected. Many are destroyed entirely. Understanding this destruction requires understanding what radiation actually is and how it interacts with biological tissue. Radiation describes energy that travels through space in the form of particles or electromagnetic waves. The word covers an enormous range of phenomena, from harmless radio waves to deadly gamma rays, from slow-moving alpha particles to relativistic heavy ions. What matters for deep space survival is ionizing radiation, energy packets powerful enough to strip electrons from atoms they encounter. The radiation relevant to deep space survival consists primarily of charged particles. Protons make up the majority of both galactic cosmic rays and solar particle events. Electrons appear in solar wind and radiation belt environments. Heavy ions, though rare, carry disproportionate biological significance. All of these particles move at velocities approaching light speed when they originate from cosmic sources. They carry enormous kinetic energy, measured in millions or billions of electron volts per nucleon. When these particles encounter matter, they transfer that energy through ionization. A charged particle passing near an atom exerts electromagnetic force on the atom's electrons. If the force is strong enough, electrons are stripped away entirely, leaving behind positively charged ions. This process repeats thousands or millions of times along the particle's path, creating a track of ionization through whatever material the particle traverses. In non-living matter, ionization causes relatively simple effects. Metals may become slightly radioactive, plastics may become brittle, electronics may malfunction as ionization disrupts semiconductor behavior. These effects are manageable through material selection and redundant system design. In living tissue, ionization triggers cascading consequences that the body struggles to contain. A single proton traveling through human tissue creates a narrow track of ionization along its path. The track diameter measures roughly 10 nanometers, smaller than most cellular organelles. Each ionization event damages or destroys molecules in its immediate vicinity. Water molecules, which comprise roughly 70% of human tissue by mass, are particularly vulnerable. When ionizing radiation strikes water, it produces highly reactive molecular fragments called free radicals. Hydroxyl radicals are especially dangerous. They survive for only microseconds, but during that time they attack whatever molecules they encounter, causing oxidative damage that can spread far beyond the original ionization track. Proteins unfold or cross-link when free radicals attack their amino acid chains. The precise three-dimensional shapes that give proteins their function are disrupted. Enzymes lose catalytic activity. Structural proteins lose mechanical strength. Receptor proteins fail to recognize their targets. Lipid membranes develop holes when free radicals attack the fatty acid chains that form cellular boundaries. The plasma membrane that separates cell interior from exterior becomes leaky. Mitochondrial membranes that maintain the electrochemical gradients powering cellular energy production become compromised. Nuclear membranes that protect genetic material from cytoplasmic enzymes become porous. Most importantly, DNA strands break. The human genome consists of roughly 3 billion base pairs organized into 46 chromosomes. This genetic information must be preserved accurately across trillions of cells and billions of cell divisions throughout a human lifetime. The molecular machinery that copies, repairs, and expresses genetic information operates with remarkable fidelity. But it evolved to handle the damage rates encountered on Earth's surface, not the damage rates found in deep space. The human body handles routine radiation damage constantly. Background radiation on Earth causes roughly 10,000 DNA lesions per cell per day. Oxidative metabolism produces free radicals that cause additional thousands of lesions. Spontaneous chemical reactions damage bases and break sugar-phosphate backbones. The genome is under constant assault simply from the process of being alive. Cellular repair mechanisms fix the vast majority of these breaks within hours. Base excision repair removes damaged nucleotides and replaces them with correct ones. Nucleotide excision repair handles larger distortions in the DNA helix. Mismatch repair corrects errors introduced during DNA replication. These systems operate continuously in every cell, scanning the genome for damage and correcting it before replication occurs. Single-strand breaks, where only one rail of the DNA double helix is severed, rarely cause permanent problems. The complementary strand provides a template for accurate repair. Enzymes recognize the break, remove damaged material, and synthesize new DNA using the intact strand as a guide. The original information is preserved because it still exists on the undamaged strand, but galactic cosmic rays are not background radiation. The damage they cause differs fundamentally from the damage that human repair systems evolved to handle. The difference lies not in the type of molecular destruction, but in its spatial concentration. Heavy ions represent the most biologically damaging component of the galactic cosmic ray flux. Iron nuclei, silicon nuclei, magnesium nuclei, calcium nuclei. These particles carry much greater charge than protons. An iron nucleus contains 26 protons compared to the single proton in hydrogen. This greater charge means greater electromagnetic interaction with atoms along the particle's path. Their ionization tracks are not narrow. They are highways of destruction that span multiple micrometers. The core of a heavy ion track experiences energy deposition thousands of times more intense than the core of a proton track. Every molecule within this core is ionized, often multiple times. The damage is not scattered randomly through the cell. It is concentrated along a linear path that may cross multiple critical structures. When a single iron nucleus traverses a cell, it does not create isolated damage sites that repair systems can address one at a time. It creates a core of complete molecular destruction surrounded by a penumbra of secondary radiation. The track core is so densely ionized that virtually every molecule along the path is destroyed. Proteins are shredded. Membranes are perforated. DNA is fragmented. DNA in this region does not suffer single-strand breaks that can be repaired using complementary strand information. It suffers clustered damage, multiple breaks occurring within one or two helical turns of the double helix. The complementary strand is destroyed along with the coding strand. There is no template for accurate repair. Clustered DNA damage presents an entirely different repair challenge. Double strand breaks, where both rails of the DNA helix are severed at nearby locations, cannot use complementary strand information for repair. The cell must use alternative mechanisms that are inherently error-prone. Two main pathways exist for double-strand break repair, and both carry significant risks of introducing permanent genetic changes. Non-homologous end joining simply glues the broken ends back together without reference to any template. This mechanism is fast, but sloppy. It frequently deletes or inserts small numbers of base pairs at the repair site. If the break occurred within a gene, the gene is likely mutated. If the break occurred in a regulatory region, gene expression patterns may be permanently altered. Homologous recombination uses a sister chromatid as a template for repair, achieving higher accuracy but requiring that the cell has recently replicated its DNA. This mechanism is slower and not always available. When it fails, cells fall back on non-homologous end, joining with its attendant error rate. When multiple double-strand breaks occur in close proximity, as happens throughout a heavy ion track, repair becomes even more problematic. The ends of different breaks can be joined incorrectly. A break on chromosome 1 might be joined to a break on chromosome 7. This creates chromosomal translocations where genetic material from different chromosomes becomes fused together. These translocations are not hypothetical possibilities. They are observed directly in astronaut blood samples. Researchers can examine lymphocytes from astronauts before and after missions and count the chromosomal abnormalities. The numbers increase predictably with mission duration. Translocations, inversions, deletions, all the signatures of error-prone double-strand break repair accumulate in the genomes of everyone who ventures into space. These repair errors are permanent. Unlike the original radiation damage, which represents broken molecular structures, repair errors represent altered information. The sequence of base pairs has changed. The arrangement of chromosomes has changed. Cell division copies these changes faithfully to daughter cells. The errors persist through every subsequent generation of cells derived from the original damaged cell. Repair errors can activate oncogenes, the genes whose uncontrolled expression promotes cancer. They can deactivate tumor suppressor genes, the genes whose normal function prevents uncontrolled cell division. They can cause cell death if critical genes are disrupted beyond function. Or, perhaps worst of all, they can cause cells to survive with genomic instability that increases error rates in subsequent generations of daughter cells. Genomic instability represents a particularly insidious consequence of heavy ion damage. A cell that survives radiation exposure with unrepaired or misrepaired DNA may function adequately for years, but its descendants show elevated mutation rates even without additional radiation exposure. The damage creates a legacy of genetic fragility that persists long after the original exposure ends. This brings us to why cumulative radiation dose matters so profoundly. Every heavy ion that traverses your body leaves permanent damage. The particle itself is gone in less than a nanosecond, but the molecular destruction it caused persists. Some cells die immediately, their damage too severe for continued function. Others accumulate mutations that alter their behavior subtly. Still others survive with chromosomal abnormalities that may not manifest as disease for years or decades. The damage does not heal between exposures, it adds up. Consider what this means for a deep space mission. On day 1, a heavy ion crosses through your liver. The cells along its path suffer clustered damage. Most die. A few survive with chromosomal translocations. On day 2, another heavy ion crosses through your lung. More cells die. More cells survive with mutations. On day 3, day 4, day 100, day 500, the process continues. Each day brings new damage. The body attempts repairs, but error-prone repair mechanisms introduce new mutations with each attempt. The cumulative burden of genetic damage grows steadily larger. Cells that escaped damage on previous days eventually receive their own heavy ion strikes. No tissue is spared, no organ system is exempt. Scientists have measured these effects in astronauts returning from space station missions. Chromosome aberrations increase with mission duration. The relationship is linear and consistent across multiple studies. More time in space equals more chromosomal damage. The slope of this relationship tells us approximately how fast genetic damage accumulates in the space radiation environment. The damage persists for years after return to Earth. Though some aberrations decline over time as damaged cells are replaced by cell division. Blood cells, which are continuously produced from stem cells in bone marrow, gradually recover their normal chromosome patterns as damaged lymphocytes die and are replaced. But other tissues with slower turnover retain radiation damage much longer. Neurons, which do not divide in adults, retain damage indefinitely. NASA researchers have found that astronauts accumulate approximately 14 additional chromosome aberrations per 100 cells per year in low Earth orbit. This baseline rate reflects the radiation environment inside Earth's magnetosphere, where the flux is lower and heavy ions are partially deflected. Deep space exposure would multiply this rate significantly. The galactic cosmic ray flux beyond Earth's magnetic field is higher. The proportion of heavy ions is greater. The biological effectiveness of each particle is increased because the magnetosphere no longer filters out the most damaging components. Estimates suggest aberration rates 3 to 10 times higher than low Earth orbit values. But chromosomal aberrations represent only one category of damage. They are relatively easy to measure, which is why they receive so much attention in astronaut studies. Other forms of damage are harder to quantify, but equally important for long-term health. Radiation also affects the cardiovascular system in ways that scientists are only beginning to understand. The heart and blood vessels seem far removed from the genetic damage that dominates discussions of radiation health effects, but mounting evidence suggests that radiation exposure promotes cardiovascular disease through mechanisms distinct from carcinogenesis. Studies of atomic bomb survivors, radiation therapy patients, and astronauts all show elevated cardiovascular disease risk following significant exposure. The effect appears at doses lower than those causing detectable cancer risk increases. It develops over decades following exposure, suggesting chronic progressive damage rather than acute injury. The mechanism appears to involve radiation damage to blood vessel walls. Endothelial cells lining arteries are exposed to every heavy ion that passes through the cardiovascular system. Damage to these cells promotes inflammation, attracts immune cells, and initiates the cascade of events that leads to atherosclerotic plaque formation. Radiation may accelerate the same processes that cause heart disease in non-irradiated populations. Apollo astronauts who traveled beyond Earth's magnetosphere show cardiovascular disease mortality rates 4 to 5 times higher than astronauts who remained in low Earth orbit. This finding comes from a very small sample size. Only 24 humans ever traveled beyond the magnetosphere, and mortality follow-up is inevitably limited. Confounding factors cannot be ruled out. Perhaps astronauts selected for lunar missions differed from other astronauts in ways that affected cardiovascular risk, but the biological mechanisms are plausible and consistent with what radiation does to vascular tissue. If deep space radiation truly accelerates cardiovascular aging, then long-duration missions face yet another pathway by which cumulative dose limits survival time. The central nervous system presents perhaps the most concerning target for space radiation damage. The brain controls everything that makes us human: cognition, memory, emotion, decision-making, personality. Damage to neural tissue threatens not just health but identity. Neurons do not divide in adults. This fundamental fact of neurobiology has profound implications for radiation effects. Other tissues repair radiation damage partly by replacing killed cells with new ones. Bone marrow produces fresh blood cells. Gut epithelium regenerates from stem cell populations. Skin replaces damaged surface cells continuously. The brain cannot do this. Damaged neurons cannot be replaced. The connections they formed with other neurons are lost permanently when they die. The memories they encoded, the skills they facilitated, the personality traits they helped create, all depend on the continued function of specific irreplaceable cells. Heavy ion radiation appears particularly effective at damaging neurons in ways that impair cognitive function. Animal studies consistently show behavioral and cognitive deficits following simulated galactic cosmic ray exposure at doses relevant to Mars missions. Mice and rats exposed to heavy ion beams show measurable changes in learning, memory, anxiety, and social behavior. The doses used in these studies match what astronauts would receive during interplanetary transit. The behavioral changes appear reliable across multiple research groups using different methodologies. The translation from rodent to human remains uncertain, but the consistency of findings raises serious concerns. Mice exposed to heavy ion radiation at Mars mission equivalent doses show impaired memory on tests requiring spatial navigation and object recognition. They show increased anxiety on tests, measuring willingness to explore novel environments. They show reduced motivation on tests requiring sustained effort for reward. They show poorer problem-solving abilities on tests requiring flexible thinking. These effects appear even when total dose is relatively low by the standards of acute radiation exposure. The mechanism seems to involve damage to the synaptic connections between neurons rather than outright cell death. Dendrites, the branching projections that receive signals from other neurons, show morphological changes following heavy ion exposure. Dendritic spines, the tiny protrusions where synaptic connections form, become less numerous and less complex. More recently, researchers discovered that heavy ion radiation reduces neurogenesis, the production of new neurons, in brain regions critical for memory formation. The hippocampus, which converts short-term memories into long-term storage, produces new neurons throughout life, in both rodents and humans. This ongoing neurogenesis appears important for certain types of learning and memory. Heavy ion exposure dramatically suppresses hippocampal neurogenesis in animal models. Fewer new neurons are produced. Those that are produced show abnormal migration and integration into existing circuits. The effects persist for months after exposure ends, suggesting permanent impairment of the brain's limited capacity for self-renewal. Heavy ion radiation also promotes neuroinflammation that persists long after exposure ends. Microglia, the brain's resident immune cells, become chronically activated following radiation exposure. They release inflammatory cytokines that damage nearby neurons. They prune synaptic connections more aggressively than normal. The brain enters a state of low-grade chronic inflammation that impairs function even without additional radiation exposure. The brain does not recover from this damage. It adapts as best it can, rerouting functions around damaged areas, compensating for lost capacity with increased effort. But cognitive function remains compromised. The astronaut who returns from a Mars mission is not the same person who left. Their brain has been physically altered by the accumulated passage of thousands of heavy ions through neural tissue. Imagine embarking on a 3-year Mars mission. For 18 months in transit, you accumulate radiation damage to your brain. Perhaps 10,000 heavy ions traverse your skull during this period. Each one leaves its track of destroyed neurons and damaged synapses. Your memory becomes less reliable. Your decision-making slows. Your emotional regulation degrades. You arrive at Mars less capable than when you left. And you face another 18 months of exposure before returning home. The radiation dose you accumulated during transit does not reset when you reach Mars. It continues contributing to cumulative damage throughout your time on the Martian surface and during your return journey. Every heavy ion track that crossed your brain left permanent changes. Every month adds more tracks. The damage accumulates whether you notice it or not. By the time you return to Earth, your cognitive function may have declined measurably from pre-mission baselines. Memory tests show deficits, reaction times have slowed, emotional stability has decreased. The mission succeeded, but you paid a permanent neurological price for the accomplishment. This brings us to cancer, the most well-documented consequence of radiation exposure, and the health endpoint that drives regulatory limits on astronaut radiation exposure. Cancer develops when a single cell accumulates enough genetic damage to escape normal growth controls. The transformed cell divides when it should not divide. It ignores signals that should trigger programmed cell death. It invades tissues where it does not belong. It recruits blood vessels to supply its metabolic needs. It spreads to distant sites throughout the body. Radiation causes exactly the type of DNA damage that drives carcinogenesis. Double-strand breaks that are misrepaired create chromosomal rearrangements. Point mutations alter protein function. Deletions remove tumor suppressor genes. Amplifications increase oncogene copy number. Every mechanism by which cancer develops can be initiated by radiation damage. The relationship between radiation dose and cancer risk is well established from decades of epidemiological research. Atomic bomb survivors have been followed for over 70 years, providing detailed information about cancer incidents across a wide range of doses. Radiation workers are monitored throughout their careers, adding data at lower dose levels. Patients receiving radiation therapy are tracked for long-term outcomes, revealing tissue-specific sensitivities. The consensus view is that cancer risk increases linearly with radiation dose, with no threshold below which risk is zero. This linear no-threshold model remains controversial among some researchers who argue that low doses may be less harmful per unit dose than high doses. But regulatory agencies worldwide accept the linear model as the best available basis for radiation protection standards. Under the linear model, every additional millisievert adds a small increment to lifetime cancer probability. For adults, the excess risk is approximately 5% per 1,000 millisieverts. A person who receives 1,000 millisieverts of radiation exposure faces roughly 5 additional chances in 100 of dying from cancer, compared to an unexposed individual. This linear no-threshold model drives regulatory limits and mission planning calculations. It implies that a Mars mission delivering 500 millisieverts increases lifetime cancer risk by roughly 2.5%. That sounds manageable when expressed as a percentage. Most people accept comparable risks from lifestyle choices like smoking or obesity, but the comparison obscures an important distinction. Lifestyle risks can be reversed. A smoker who quits reduces their cancer risk over time. An obese person who loses weight improves their health outcomes. Radiation exposure cannot be reversed. The DNA damage is permanent. The mutations persist in surviving cells. The elevated cancer risk remains for life. The astronaut who completes a Mars mission carries elevated cancer risk for the rest of their life. They cannot undo the exposure. They cannot repair all the DNA damage their cells sustained. Their cells carry mutations that may or may not manifest as disease. The dice have been loaded against them, and every year that passes represents another roll, and cancer risk does not capture the full picture of radiation harm. Cataracts develop at lower doses than most cancers. Radiation damages the lens of the eye, causing protein aggregation that eventually clouds vision. The lens consists of specialized cells that are never replaced once formed. Damage accumulates throughout life, as new lens fibers are added at the periphery, while old fibers remain in the center. Many astronauts develop cataracts earlier than expected, based on their age and other risk factors. The excess risk appears most clearly for astronauts with longer flight times and higher estimated radiation exposures. Cataracts are surgically treatable, which reduces their significance for overall health. But they represent yet another manifestation of cumulative radiation damage. Immune function degrades following radiation exposure. T cells are particularly radiosensitive, showing reduced counts and altered function following radiation damage. The immune system protects against infections and surveys the body for cancerous cells. Impaired immunity could compound other radiation effects by reducing the body's capacity to destroy transformed cells before they establish tumors. Studies of astronauts show altered immune cell populations and reduced immune response following long-duration missions. Latent viruses reactivate more frequently. Immune markers show patterns consistent with premature immunological aging. Fertility may be affected by radiation exposure. Gonads are among the most radiosensitive tissues in the human body. The rapidly dividing cells that produce sperm are easily killed by radiation. The oocytes that comprise a woman's lifetime supply of eggs are vulnerable to damage even though they do not divide in adults. Radiation damage to reproductive cells can cause temporary or permanent infertility. Even if fertility is preserved, radiation may increase mutation rates in offspring by damaging DNA in germ cells. This concern has limited practical significance for astronauts, who are typically beyond reproductive age by the time they complete long-duration missions, but it represents another tissue system vulnerable to cumulative dose effects. Each of these effects follows from the same underlying mechanism. Radiation deposits energy in tissue, causing ionization, which damages molecules, which disrupts cellular function. More radiation means more ionization means more damage. The relationship is not mysterious. It follows directly from physics and biochemistry. The dose determines the damage. The damage accumulates. The consequences manifest over time. This brings us back to the fundamental constraint governing deep space survival. Every minute spent beyond Earth's protection adds to cumulative radiation dose. Every millisievert accumulated increases the probability and severity of health consequences. Every heavy ion that traverses the body leaves permanent traces in the form of dead cells, mutated survivors, and misrepaired chromosomes. There is no recovery period that erases previous exposure. An astronaut who returns from one deep space mission and rests for 5 years before attempting another does not begin the second mission with a clean slate. The damage from the first mission persists in every cell that survived the exposure. The second mission adds to this existing burden rather than starting fresh. There is no adaptation that makes subsequent exposure less harmful. The body does not become radiation resistant through experience. If anything, accumulated damage may make tissues more vulnerable to additional insults. Cells with unrepaired damage may be more likely to undergo malignant transformation when hit again. The damage simply adds up. The question facing mission planners is not whether to accept radiation harm. That decision was made when they chose to leave Earth. Every deep space mission imposes radiation costs on its crew. The question is how much harm to accept and how to distribute it across crew members and missions. But no amount of clever mission design changes what radiation does to human tissue. No operational procedure prevents heavy ions from causing clustered DNA damage. No crew selection criterion identifies humans who are immune to radiation effects. All humans are vulnerable. All humans accumulate damage at roughly similar rates. Variation exists in individual radiosensitivity, but no one escapes the fundamental physics of particle interactions in tissue. The biology is clear. The physics is clear. What remains unclear is whether any technology can change the equation enough to allow humans to survive long-duration, deep space missions without accumulating damage that shortens lives, impairs function, and ultimately proves fatal. That question leads directly to examining every proposed solution for protecting astronauts from space radiation. Shielding, propulsion, pharmaceuticals, genetic modification. Each approach has been studied extensively. Each faces fundamental limitations that prevent it from solving the cumulative dose problem. As we will see, each solution either fails entirely or reduces dose rates by insufficient margins to change the fundamental conclusion. The mechanisms of destruction operate regardless of human ingenuity. The particles keep coming. The damage keeps accumulating. The body keeps failing to fully repair what radiation breaks. The obvious solution to space radiation is shielding. Put enough material between astronauts and the radiation source, and the particles will be stopped before reaching human tissue. This approach works beautifully for many radiation hazards on Earth. Radiologists stand behind lead screens when operating X-ray machines. Nuclear plant workers shelter behind concrete walls meters thick. Hospital patients receive radiation therapy through carefully shaped beams that spare healthy tissue by simple geometric blocking. Surely some material configuration can shield against galactic cosmic rays. The physics says otherwise. And understanding why reveals one of the most frustrating constraints facing deep space exploration. Galactic cosmic rays are not X-rays or gamma rays. The distinction matters enormously for shielding effectiveness. X-rays and gamma rays are electromagnetic radiation, massless photons that interact with matter through well-understood processes. When a photon encounters an atom, it may be absorbed entirely, transferring all its energy to an electron, or it may scatter, changing direction while losing some energy. Either way, the photon is gone. It does not produce new photons traveling in different directions. Dense materials like lead are excellent photon absorbers because their tightly packed atoms present many opportunities for these interactions. Galactic cosmic rays are massive charged particles traveling at relativistic velocities. They consist of atomic nuclei, from single protons to iron nuclei containing 26 protons and roughly 30 neutrons. These particles carry mass and momentum in addition to kinetic energy. When they encounter matter, they do not simply disappear. They interact with atomic nuclei in the shielding material through processes governed by nuclear physics rather than atomic physics. The interaction that dominates shielding effectiveness is called nuclear fragmentation. When a high-energy heavy ion collides with an atomic nucleus in shielding material, both nuclei can shatter into smaller fragments. The collision does not absorb the incoming particle cleanly. It converts one dangerous particle into multiple dangerous particles traveling in various directions. Consider what happens when an iron nucleus in a galactic cosmic ray strikes an aluminum nucleus in spacecraft hull material. The iron nucleus carries 56 nucleons bound together by strong nuclear forces. The aluminum nucleus carries 27 nucleons similarly bound. When these nuclei collide at a significant fraction of light speed, the binding forces are overwhelmed. Both nuclei fragment. The fragments spray outward with high energy. Some are neutrons, which carry no charge and therefore pass through matter easily. Others are protons, which behave like lower energy versions of the original cosmic ray. Still others are lighter ions like helium nuclei, lithium nuclei, beryllium nuclei. Each fragment can cause its own biological damage if it reaches human tissue. This secondary radiation problem creates a counterintuitive result. Thin shielding can actually increase radiation dose to astronauts rather than decrease it. The material stops some primary particles, but generates secondary particles that would not otherwise exist. The secondaries may carry less energy individually, but there are more of them, and their combined biological effect can exceed the original particle's impact. Studies conducted at particle accelerator facilities have consistently demonstrated this effect. Researchers expose biological samples and radiation detectors to simulated galactic cosmic ray beams with various shielding configurations between source and target. Aluminum shielding, the standard material for spacecraft hulls, provides minimal protection against heavy ions and can be counterproductive at certain thicknesses. The worst thickness range falls between roughly 5 and 20 grams per square centimeter for aluminum. Thinner shielding does little. Thicker shielding begins to show benefit. But even very thick aluminum shielding fails to reduce dose rates to levels compatible with long-duration missions because the secondary particle cascade continues producing harmful radiation throughout the shielding depth. Different materials produce different secondary particle spectra. The key variable is the atomic mass of the shielding nuclei. Heavy elements like lead, excellent for photon shielding, perform poorly against cosmic rays because their large nuclei fragment into many secondaries when struck. Light elements perform better because their small nuclei produce fewer fragments and the fragments carry less total energy. Hydrogen represents the optimal shielding element from a nuclear physics perspective. A hydrogen nucleus is simply a proton. When a cosmic ray strikes a hydrogen nucleus, the collision cannot produce nuclear fragmentation because there is nothing to fragment into. The cosmic ray may lose energy through electromagnetic interactions, but it does not spawn a cascade of secondary particles. This insight led researchers to evaluate hydrogen-rich materials for spacecraft shielding. Water consists of 2 hydrogen atoms for every oxygen atom, giving it a higher hydrogen density than any practical structural material. Water walls around crew quarters could reduce galactic cosmic ray dose by perhaps 20 to 30% compared to equivalent mass of aluminum shielding. But water is heavy. A meaningful water shield would require layers at least 30 centimeters thick surrounding the crew habitat. For a habitat volume large enough to house a crew for months or years, this translates to tens of thousands of kilograms of water mass. Every kilogram launched from Earth costs thousands of dollars and requires proportionally more propellant. A water-shielded Mars mission would require launch capabilities far exceeding current or near-term systems. The mass penalty cascades through mission design. Heavier spacecraft require more propellant. More propellant requires larger tanks. Larger tanks require stronger structures. Stronger structures add more mass. The rocket equation, which governs the relationship between payload mass and propellant requirements, punishes additional mass exponentially. Doubling payload mass more than doubles the propellant needed to achieve the same trajectory. Polyethylene and other hydrogen-rich plastics show similar shielding benefits to water with somewhat easier handling characteristics. High-density polyethylene contains roughly 14% hydrogen by mass, less than water's 11%, but still far better than aluminum. NASA has tested polyethylene shielding extensively in ground-based experiments and limited spaceflight applications. Polyethylene reduces dose rates more effectively than aluminum per unit mass. It can be fabricated into structural panels that serve dual purposes as radiation shielding and meteorite protection. Several spacecraft designs have incorporated polyethylene-based shielding for crew quarters. The International Space Station uses polyethylene tiles in sleeping compartments to reduce astronaut exposure during their most stationary hours. But the reductions remain insufficient to enable long-duration missions beyond Earth's magnetosphere. Here is the problem quantified with specific numbers that illuminate the gap between what shielding can achieve and what mission safety requires. In interplanetary space, galactic cosmic rays deliver approximately 400 to 900 millisieverts per year depending on solar cycle conditions. This range reflects the modulation of cosmic ray flux by solar magnetic activity. During solar maximum, the expanded solar magnetic field deflects more cosmic rays, reducing the flux reaching Earth's orbital distance. During solar minimum, cosmic ray intensity increases as the solar magnetic barrier weakens. With optimized polyethylene shielding of practical thickness, perhaps 10 to 20 grams per square centimeter distributed appropriately around crew quarters, dose rates might decrease to 300 to 700 millisieverts per year. The reduction is meaningful, but far from sufficient. Astronauts would still accumulate hundreds of millisieverts annually, exceeding safe exposure rates by large margins. A 3-year Mars mission under these optimistic shielding conditions would deliver 900 to 2,100 millisieverts total. The lower bound approaches NASA career limits for the oldest male astronauts. The upper bound exceeds career limits for everyone, and these estimates assume no major solar particle events occur during the mission, which would add significant additional exposure despite the availability of storm shelters. The mathematics is unforgiving. Even the best practical shielding reduces dose rates by only 30 to 50%. Total exposure still mounts at rates measured in hundreds of millisieverts per year. Career limits are still reached or exceeded within years rather than decades. The shielding helps, but it does not solve the fundamental problem. This brings us to a crucial realization about the limits of passive shielding technology. Passive shielding cannot solve the galactic cosmic ray problem because no practical shield thickness stops enough primary particles while avoiding excessive secondary particle production. There exists no material that absorbs heavy ions without producing secondaries. There exists no material configuration that reduces dose rates to levels compatible with indefinite human presence in deep space. The fundamental physics of nuclear fragmentation sets limits that material engineering cannot overcome. You can optimize material selection, you can arrange shielding layers strategically, you can position crew quarters within spacecraft to maximize the shielding provided by other systems. But you cannot change the nuclear physics that governs how cosmic rays interact with matter. Some researchers have proposed using lunar or Martian regolith as shielding material for surface habitats. The concept is appealing because it uses locally available resources rather than materials launched from Earth. Bury a habitat under several meters of regolith and cosmic ray exposure would decrease substantially. Regolith shielding faces its own challenges. Lunar regolith consists primarily of silicate minerals with average atomic masses much higher than hydrogen-rich materials. It produces more secondaries per incident cosmic ray than polyethylene would. Achieving equivalent protection requires thicker layers and correspondingly more excavation and construction effort. Mars atmospheric shielding provides roughly 1/4 the protection of Earth's atmosphere. The Martian surface receives less radiation than free space, but still far more than Earth's surface. Burying habitats under Martian regolith could reduce exposure further, but the engineering requirements for excavating and constructing such shelters remain formidable. Neither approach addresses the transit radiation problem. Astronauts must still travel between Earth and their destination through unshielded interplanetary space. Surface shielding protects them after arrival, but does nothing during the months of transit that constitute the majority of mission radiation exposure. Some researchers have proposed active magnetic shielding systems that would deflect charged particles before they reach the spacecraft. This approach mimics Earth's magnetosphere, creating an artificial magnetic cocoon around the habitat. Rather than absorbing cosmic rays and dealing with secondary particles, magnetic shielding would deflect primaries away from the protected volume entirely. The concept is elegant. Charged particles curve when passing through magnetic fields. The radius of curvature depends on particle momentum and field strength. Strong enough fields over large enough distances could bend cosmic ray trajectories enough to miss the spacecraft entirely. No interaction with shielding material means no secondary particle production. The engineering is essentially impossible with current technology. To deflect galactic cosmic ray particles, which carry enormous momentum due to their relativistic velocities and substantial masses, magnetic fields must be extraordinarily strong and must extend over large distances. Calculations suggest field strengths of several Tesla over regions spanning tens of meters would be required to deflect the most energetic and most biologically damaging components of the cosmic ray flux. For comparison, MRI machines generate fields of roughly 1.5 to 3 Tesla within bores, measuring less than 1 meter in diameter. The total magnetic energy stored in an MRI system is substantial but manageable. Scaling similar field strengths to the volumes required for spacecraft protection would increase stored magnetic energy by factors of thousands or millions. Generating such fields requires superconducting magnets, operating at cryogenic temperatures. Normal conducting electromagnets would require impossibly large power supplies and would generate waste heat exceeding any spacecraft's thermal management capacity. Only superconductors, which carry current without resistance, can sustain the required field strengths for extended periods. But superconducting magnets require cooling to temperatures near absolute zero. In space, maintaining cryogenic temperatures requires active refrigeration systems that consume significant power and add substantial mass. The refrigeration systems must operate continuously for years without maintenance. Any failure that allows the magnets to warm would result in catastrophic quench events where stored magnetic energy converts suddenly to heat. Several magnetic shielding concepts have been studied over the decades. Toroidal field configurations would create a donut-shaped magnetic region around the spacecraft, deflecting particles inward or outward depending on their charge. Confined plasma clouds would use ionized gas to carry currents that generate protective fields without solid conductors. Electrostatic deflection systems would use high voltages rather than magnetic fields to repel charged particles. None have progressed beyond conceptual design and laboratory-scale demonstration. The mass, power, and complexity requirements exceed what any near-term mission architecture can support. The engineering challenges of building, launching, deploying, and maintaining superconducting magnet systems over multi-year missions remain formidable. Even optimistic projections suggest that magnetic shielding might reduce dose rates by 50%, while adding thousands of kilograms to spacecraft mass. More realistic assessments suggest that practical magnetic systems would provide less protection than equivalent masses of passive shielding, while introducing failure modes that could leave crews completely unprotected. No magnetic shielding system has been tested in space at any meaningful scale. No magnetic shielding system has been demonstrated at relevant scales on Earth. The technology readiness level remains low despite decades of conceptual studies. Magnetic shielding represents a theoretical possibility rather than a practical solution for any mission planned within the next several decades. Electrostatic shielding faces similar fundamental obstacles. Charged particles can be deflected by electric fields as well as magnetic fields. A spacecraft surrounded by a highly charged shell would repel particles of the same charge sign. Since cosmic rays are predominantly positively charged nuclei, a positively charged spacecraft would deflect them away from the protected volume. The voltages required are staggering. Deflecting relativistic heavy ions requires electric potentials measured in billions of volts. Maintaining such potentials in the space environment, where solar wind plasma constantly bombards surfaces and tends to neutralize charge accumulations, requires continuous power input. The electric fields around such a highly charged spacecraft would create their own hazards for crew members attempting extravehicular activities. No electrostatic shielding system has progressed beyond theoretical analysis. The engineering challenges exceed those facing magnetic approaches without offering superior protection. Electrostatic concepts have largely been abandoned in favor of continued research on magnetic systems and passive material shielding. Pharmaceutical interventions represent a fundamentally different approach to the radiation problem. Rather than blocking particles before they reach tissue, pharmaceutical approaches would reduce the biological damage caused by particles that do reach tissue. If radiation damage cannot be prevented, perhaps it can be mitigated through drugs that protect tissues or enhance repair mechanisms. Radioprotective compounds do exist and have been studied extensively for military and medical applications. The most effective agents are sulfhydryl compounds that scavenge free radicals before they can attack cellular macromolecules. Amifostine, approved for reducing side effects in cancer patients receiving radiation therapy, can reduce radiation damage to healthy tissues by roughly 30% when administered shortly before exposure, but these compounds have significant side effects that preclude long-term use. Amifostine causes nausea, vomiting, and hypotension in many patients. It must be administered within 30 minutes of radiation exposure to be effective, making it useless against the continuous low-dose exposure from galactic cosmic rays. Its protective effect lasts only hours, meaning daily or more frequent dosing would be required for chronic exposure scenarios. Taking radioprotective drugs continuously for a 3-year mission is not feasible with current pharmaceuticals. The side effects would impair crew performance and quality of life. The protective benefit would be partial at best. The total exposure would still mount, just slightly more slowly. More promising research focuses on enhancing the body's natural DNA repair mechanisms rather than preventing damage in the first place. Some compounds appear to accelerate repair of double-strand breaks by upregulating key repair enzymes. Others reduce error rates in repair processes by promoting the more accurate homologous recombination pathway over error-prone non-homologous end joining. These approaches show promise in laboratory studies. Mice treated with certain compounds before radiation exposure show fewer chromosome aberrations and better survival rates than untreated animals receiving equivalent doses. Human clinical trials have explored similar compounds for protecting cancer patients from radiation therapy side effects. But mitigation is not prevention. Reducing damage by 30% still leaves 70% of the damage occurring. The running total of exposure still climbs with each passing day in deep space. The mutations still occur with each particle that traverses cell nuclei. The long-term consequences are reduced but not eliminated. Even a highly effective pharmaceutical intervention that halved all radiation damage would only double the time astronauts could spend in deep space before reaching equivalent health outcomes. Career limits would be extended from years to perhaps a decade. Long-duration missions would become somewhat more feasible, but the fundamental constraint would remain. Exposure mounts. Time runs out. The arithmetic changes quantitatively, but not qualitatively. Other pharmaceutical research targets specific organ systems, particularly vulnerable to radiation damage. Anti-inflammatory compounds might reduce the neuroinflammation that causes cognitive deficits following heavy ion exposure. Cardiovascular protective agents might slow the accelerated atherosclerosis observed in irradiated populations. Antioxidants might reduce oxidative stress that persists long after primary radiation damage occurs. Each of these approaches addresses real biological mechanisms that contribute to radiation harm. Each might provide meaningful benefit to astronauts on deep space missions. None changes the fundamental relationship between total exposure and health outcomes. The particles still cause damage. The damage still compounds over time. The consequences still manifest eventually. Genetic modification has been proposed as a more radical intervention. Some organisms possess extraordinary radiation resistance that far exceeds anything found in mammals. The bacterium Deinococcus radiodurans survives radiation doses thousands of times higher than those fatal to humans. It can reassemble its genome from hundreds of double-strand breaks that would kill any human cell. The mechanisms underlying this remarkable resistance are partially understood. Deinococcus maintains multiple redundant copies of its genome, providing templates for accurate repair even when all copies are damaged. It possesses highly efficient repair enzymes that locate and rejoin broken DNA, ends with exceptional speed and accuracy. It produces specialized proteins that protect DNA from damage during the repair process and prevent chromosome fragmentation. Could humans be engineered with similar capabilities? The scientific barriers to such modification are immense. Human radiation sensitivity results from fundamental aspects of our cellular biology that evolved over hundreds of millions of years. Our DNA repair pathways interact with cell cycle controls, apoptosis mechanisms, and tumor suppression systems in complex ways that we do not fully understand. Altering repair efficiency could have unpredictable effects on these interconnected systems. DNA repair pathways interact particularly closely with cancer suppression mechanisms. Genes like p53 that promote cell death in response to DNA damage also serve as critical tumor suppressors. Modifications that allowed cells to survive more radiation damage might simultaneously allow damaged cells to survive and proliferate when they should die. Reducing radiation harm might increase spontaneous cancer risk, even in the absence of radiation exposure. The genetic modifications that confer radiation resistance in bacteria may simply not be transferable to human biology. Bacterial cells have fundamentally different organization than human cells. They lack nuclei, lack complex chromosome structures, lack the elaborate regulatory systems that govern gene expression in multicellular organisms. What works in Deinococcus may have no equivalent application in human tissue. Even if such modifications were possible and safe, they would not eliminate radiation damage. They would only change the balance between damage and repair. Heavy ion tracks through neurons would still disrupt synaptic connections because the physical destruction occurs regardless of repair capability. Cardiovascular damage would still build up because blood vessel walls cannot be repaired to pristine condition. The biological cost would still be paid in ways that repair mechanisms could only partially address. Genetic modification remains speculative for radiation protection purposes. No human gene therapy has ever been approved for enhancing radiation resistance. No animal studies have demonstrated meaningful protection through genetic modification of repair pathways. The concept remains theoretical and faces ethical objections beyond purely scientific challenges. This persistent failure of every proposed solution reveals something fundamental about the radiation protection problem. Every approach attacks a symptom rather than the underlying cause. Shielding tries to block particles but cannot prevent secondary production or stop the highest energy primaries. Drugs try to reduce damage but cannot prevent the molecular destruction caused by ionizing radiation. Genetic modification tries to enhance repair but cannot restore destroyed neurons or reverse cardiovascular aging. Magnetic fields try to deflect particles but cannot be generated with sufficient strength and extent using foreseeable technology. None address the root constraint that total radiation exposure grows continuously without practical upper limits. The only way to reduce total dose is to reduce either the dose rate or the exposure time. Reducing dose rate requires shielding or avoidance, approaches that face the limitations already described. Reducing exposure time requires faster travel between Earth and destinations in deep space. Faster propulsion systems would help enormously. The longer astronauts spend in interplanetary space, the more radiation they absorb. Cut transit time in half, and you cut transit dose in half. Cut transit time by a factor of 4, and career limits might accommodate round-trip missions to Mars with margin to spare. Chemical rockets, the only propulsion technology with extensive human spaceflight heritage, achieve specific impulses around 450 seconds. This limits transit times to Mars to roughly 7 to 9 months each way, depending on trajectory and launch window. 18 months of transit exposure dominates total mission dose, regardless of surface stay duration. Nuclear thermal propulsion offers roughly double the specific impulse of chemical rockets. Transit times to Mars could decrease to 4 or 5 months with optimized trajectories. The dose reduction would be meaningful but not transformative. Astronauts would still receive hundreds of millisieverts during transit phases. Nuclear thermal engines have been designed and ground-tested but never flown. The NERVA program in the 1960s demonstrated the technical feasibility of nuclear thermal propulsion before being cancelled for budgetary and political reasons. Reviving this technology would require substantial investment and would face regulatory hurdles for launching nuclear materials. More advanced propulsion concepts promise greater specific impulse and correspondingly shorter transit times. Nuclear pulse propulsion, using repeated nuclear explosions to accelerate spacecraft, could theoretically achieve specific impulses in the thousands of seconds. Fusion propulsion, if the engineering challenges of sustained fusion reactions can be solved, could achieve similar or better performance. These technologies remain speculative. No nuclear pulse propulsion system has ever been tested at any scale. No fusion propulsion system exists, even in prototype form. The timelines for developing such capabilities, if they prove feasible at all, span decades rather than years. Shorter transit times would reduce total exposure, but would not eliminate it. A crew that reaches Mars in 2 months still absorbs 50 to 100 millisieverts during transit. Time on the Martian surface adds more exposure at rates of roughly 20 millisieverts per month. The return trip adds still more. Career limits are approached or exceeded, even with dramatically faster travel times, if surface stays extend beyond a few weeks. And propulsion improvements have their own massive engineering challenges, independent of radiation considerations. Higher performance propulsion systems require more advanced materials, more sophisticated controls, more challenging testing programs. Each alternative propulsion system trades radiation exposure, time for other technical risks, and development costs. There is no escape from the fundamental trade-off. You can shield more and travel heavier, accepting the mass penalties and launch cost increases that thicker shielding entails. You can travel faster and burn more propellant, if propulsion technology advances sufficiently to enable higher velocities. You can accept more biological risk and push career limits higher, acknowledging that astronauts will face elevated probabilities of cancer, cardiovascular disease, and cognitive decline. But the radiation dose still grows according to physics that engineering cannot change. Every proposed solution reduces the rate of growth or extends the tolerable total. None removes the constraint that total dose ultimately limits mission duration and human survival time in deep space. This brings us back to the central constraint that governs all deep space mission planning. No foreseeable combination of shielding, propulsion, and pharmaceutical protection can enable humans to survive indefinitely in deep space. The galactic cosmic ray flux imposes a baseline dose rate that cannot be reduced below dangerous levels using any technology demonstrated or credibly projected for development within the coming decades. This dose grows continuously, regardless of what countermeasures are employed. It causes progressive, irreversible damage to every organ system in the human body. The damage manifests as elevated cancer risk, accelerated cardiovascular aging, cognitive decline, immune suppression, and reduced fertility. Each consequence follows from the fundamental interaction between ionizing radiation and biological tissue. The shielding paradox is not that we lack adequate materials. Engineers have access to every element in the periodic table and can fabricate materials with precisely controlled compositions and structures. The paradox is that the physics of particle interactions makes adequate shielding impossible with any foreseeable technology. Nuclear fragmentation produces secondaries regardless of material selection. Magnetic fields cannot be generated with sufficient strength regardless of engineering investment. Understanding this constraint allows us to calculate exactly how long humans can survive under various deep space scenarios. Those calculations, grounded in measured dose rates and established biological response relationships, reveal the true boundaries of human presence in the cosmos. The numbers are not encouraging, but they represent physical reality rather than engineering limitations that future innovation might overcome. The mathematics of survival in deep space reduces to a simple equation. Cumulative dose equals dose rate multiplied by time. This relationship governs everything. Mission duration is limited by how quickly accumulated dose approaches the threshold, where consequences become unacceptable. Every scenario, every destination, every proposed technology must be evaluated against this fundamental constraint. The equation contains no loopholes. It offers no exceptions for human ambition or technological cleverness. It simply describes how radiation exposure accumulates in biological tissue, regardless of what countermeasures are employed or what dreams motivate the journey. Let us apply this equation systematically to every destination humans have proposed visiting or settling. The results will illuminate the true boundaries of human presence in the cosmos. Begin with the Moon, the nearest deep space destination and the only extraterrestrial body humans have ever walked upon. The lunar surface lies beyond Earth's magnetic field. The magnetosphere's protective influence ends roughly 60,000 kilometers from Earth, and the Moon orbits at an average distance of 384,000 kilometers. Astronauts on the lunar surface receive the full galactic cosmic ray flux, modulated only by solar activity and whatever shielding their habitat provides. Surface dose rates on the Moon average approximately 380 millisieverts per year. This figure comes from measurements made by instruments aboard lunar orbiters and surface missions, cross-checked against theoretical calculations of cosmic ray flux at Earth's orbital distance. The actual rate varies significantly depending on solar cycle phase, ranging from roughly 300 millisieverts during solar maximum to nearly 500 millisieverts during solar minimum. The lunar surface provides one significant advantage over free space. The Moon itself blocks radiation from below, reducing exposure by roughly half compared to an astronaut floating in interplanetary space with radiation arriving from all directions. This geometric shielding is automatic and requires no engineering effort. It represents the minimum protection any lunar surface mission would provide. With modest additional shielding from regolith covering or habitat materials, dose rates might decrease further to 250 to 300 millisieverts per year. Burying habitats under several meters of lunar soil would provide more substantial protection, potentially reducing doses to 150 to 200 millisieverts annually. Such construction would require significant excavation capability, but uses only locally available materials. Under the most optimistic shielding conditions achievable with near-term technology, astronauts could remain on the lunar surface for roughly 3 to 4 years before reaching NASA career exposure limits. More realistically, with modest surface habitats providing limited shielding, career limits would be reached in 2 to 3 years of continuous occupancy. Solar particle events would require shelter throughout any extended lunar stay. The warning time for solar events, typically 15 minutes to several hours, would allow crews to reach protected areas before the particle flux arrives. Storm shelters with heavy shielding could reduce event exposure to manageable levels, but the cumulative dose from galactic cosmic rays, which cannot be sheltered against effectively, determines the ultimate time limit regardless of how well crews weather individual storms. This analysis suggests that permanent lunar bases are not impossible with current understanding of radiation, biology, and protection technology. They are achievable within certain constraints. The constraints are severe. No individual could spend decades on the Moon without exceeding safe radiation exposure by large margins. Crew rotation would be mandatory, with each astronaut contributing perhaps 2 to 4 years of lunar service before returning to Earth permanently. Each returning crew member would carry elevated cancer risk, accumulated cardiovascular damage, and whatever cognitive effects their exposure produced. The Moon represents the best-case scenario for sustained deep space human presence, close enough for rapid resupply and emergency return, with transit times measured in days rather than months. accessible with propulsion technology that has been demonstrated and refined over decades, surface materials available for shielding construction without launching mass from Earth, a destination where humans have already proven they can survive, at least briefly, and still, cumulative radiation limits occupancy to years rather than lifetimes. The Moon is reachable. It is not permanently habitable in the sense that Earth is habitable. Human presence there will always be temporary, constrained by the ticking clock of accumulated dose. Mars presents a more challenging scenario that illustrates how the cumulative dose equation scales with distance and transit time. The transit to Mars takes roughly 9 months each way, with conventional chemical propulsion. The precise duration depends on launch window selection, trajectory optimization, and propulsion system performance, but 7 to 9 months represents a reasonable planning figure for the technology currently available or under development. During this transit period, astronauts are fully exposed to the interplanetary radiation environment. Unlike the lunar surface, where the destination body provides geometric shielding from below, interplanetary space offers radiation exposure from all directions simultaneously. No planetary body provides partial shielding during transit. No emergency return is possible once the spacecraft commits to its trajectory. The radiation dose accumulates continuously with no possibility of mitigation beyond whatever shielding the spacecraft provides. Transit dose alone, averaging the galactic cosmic ray exposure over 18 months of deep space travel for a round-trip mission, amounts to 300 to 900 millisieverts, depending on solar conditions and shielding effectiveness. This estimate comes from measurements made by the Radiation Assessment Detector aboard the Mars Science Laboratory during its cruise to Mars in 2011 and 2012, extrapolated to longer durations and varying solar conditions. The range is wide because solar cycle timing dramatically affects cosmic ray flux. A mission launched during solar maximum would experience lower galactic cosmic ray intensity, but higher solar particle event risk. A mission launched during solar minimum would face the opposite trade-off. Neither timing offers a clearly superior option from a total dose perspective. This transit dose alone represents roughly 25 to 65% of career limits before astronauts even arrive at their destination. They have not yet begun surface operations. They have not yet contributed to mission science objectives. They have simply traveled through space, accumulating dose with every passing hour, approaching limits that will constrain everything they do afterward. The Martian surface offers less protection than Earth, but substantially more than open space. The thin Martian atmosphere, though less than 1% as dense as Earth's atmosphere, blocks some radiation through interactions with atmospheric molecules. The planet itself provides hemispherical shielding, just as the Moon does. Surface dose rates on Mars are approximately 230 to 270 millisieverts per year, roughly half the free space rate but still enormously elevated compared to Earth's surface. Measurements from the Curiosity rover's radiation detector confirm these estimates and reveal the daily and seasonal variations in surface radiation environment. A minimal Mars mission with 30 days of surface operations following 9 months outbound transit would accumulate roughly 300 to 750 millisieverts total before the return journey even begins. Add another 9 months of transit exposure for the return, and total mission dose reaches 450 to 1,100 millisieverts. This approaches or exceeds career limits for many crew members. It means the first humans on Mars will almost certainly never return to deep space. Their single mission will consume their entire lifetime radiation allowance, and then some. They will return to Earth as radiation casualties who successfully completed their mission but paid a permanent biological price for the accomplishment. Extended Mars surface stays of 1 to 2 years, which mission architectures typically require for favorable orbital mechanics and meaningful scientific productivity, push cumulative doses still higher. 18 months of transit exposure plus 18 months of surface exposure yields total doses of 500 to 1,200 millisieverts. These missions exhaust or exceed career limits for all crew members regardless of age or sex. Every astronaut who completes an extended Mars surface mission will have accumulated radiation exposure, approaching or exceeding the threshold NASA considers acceptable for an entire career. None will have margin remaining for subsequent missions. All will face elevated health risks for the remainder of their lives. Mars colonization, the permanent settlement of humans on another planet that visionaries have advocated for decades, requires accepting radiation exposure far beyond any current safety standard. Colonists would not be visiting Mars and returning. They would be living there indefinitely, accumulating dose year after year without respite. Under the best achievable shielding conditions, colonists would still accumulate 150 to 250 millisieverts annually throughout their lives on Mars. Over a 30-year period, this amounts to 4,500 to 7,500 millisieverts of cumulative exposure. Over a normal human lifespan, doses would exceed 10,000 millisieverts. These are not theoretical concerns. They translate directly to health outcomes. Cancer rates among Mars colonists would be dramatically elevated compared to Earth populations. The linear no-threshold model predicts excess cancer mortality of roughly 5% per 1,000 millisieverts. A colonist accumulating 5,000 millisieverts of lifetime exposure would face approximately 25% additional probability of dying from radiation-induced cancer beyond their baseline cancer risk. Cognitive function would progressively decline as heavy ion damage accumulated in neural tissue. The effects documented in animal studies, memory impairment, reduced motivation, increased anxiety, slower decision-making, would manifest in human colonists over years of continuous exposure. The founding generation might retain adequate function. Their children and grandchildren would show progressively worse outcomes. Cardiovascular systems would age prematurely as radiation damage accumulated in blood vessel walls. Heart disease and stroke rates would exceed Earth populations by substantial margins. Life expectancy on Mars would be shorter than life expectancy on Earth, even accounting for other hazards of the Martian environment. Some advocates argue that these risks are acceptable for pioneers willing to sacrifice their health for human expansion beyond Earth. They invoke historical analogies to explorers who face dangers and early death to open new frontiers. They suggest that sufficiently motivated volunteers could consent to radiation risks that would be unacceptable for ordinary astronauts. But the risks extend beyond the pioneers themselves. This is where the ethical implications become most troubling. Children born on Mars would begin accumulating radiation dose from their first breath. Unlike adult colonists who arrive with fully developed organ systems, children would experience radiation exposure during critical developmental periods when tissues are most vulnerable. Developing brains are far more radiosensitive than adult brains. Neurogenesis occurs rapidly during childhood, creating new neurons that heavy ion radiation would damage or destroy. Cognitive development would occur under constant radiation bombardment that animal studies suggest impairs learning, memory, and emotional regulation. Developing immune systems, cardiovascular systems, and reproductive systems would all experience elevated damage rates compared to adult tissues. Cancer risk models suggest that childhood radiation exposure carries higher lifetime risk per unit dose than adult exposure, because children have more years during which radiation-induced cancers can develop. The first generation born on Mars would face lifetime cancer risks far exceeding their parents. Their neurological development would be compromised by an environment their brains did not evolve to tolerate. Their life expectancy would be shorter than their parents, who at least began life on Earth with undamaged tissues, and their children would face the same hazards, compounded by whatever genetic damage their parents' germ cells accumulated before conception. The multi-generational consequences of permanent Mars settlement extend beyond individual health outcomes to population-level effects that could threaten the viability of any colony attempting to sustain itself across generations. Are future generations, who cannot consent, being condemned to shortened lives and elevated disease rates so that their ancestors could fulfill a dream of planetary expansion? This question has no easy answer, but it cannot be avoided by those who advocate permanent Mars colonies. The radiation environment is not a problem that future technology will solve for children born before that technology exists. Destinations beyond Mars face even starker limitations that close the door on human presence entirely. The asteroid belt lies roughly 2.5 to 4 astronomical units from the Sun, requiring transit times of 18 months to several years with current propulsion technology. Mining operations or scientific stations in the belt would expose personnel to continuous galactic cosmic ray flux with no nearby planetary body, providing even partial shielding. Asteroid surface operations would offer minimal protection. Most asteroids are too small to provide significant geometric shielding. Their surfaces are exposed to radiation from nearly all directions, just as interplanetary space is. Burying habitats beneath asteroid regolith might provide some protection, but the engineering requirements for excavating and constructing such facilities on low-gravity bodies remain formidable. Cumulative doses for asteroid belt operations would exceed Mars mission doses substantially due to longer transit times and reduced surface shielding options. Career limits would be exhausted during outbound transit alone for most realistic mission architectures. The asteroid belt is accessible to robotic probes but not to humans under any credible near-term scenario. Jupiter's moons present particular challenges that go beyond galactic cosmic ray exposure. The Jovian system is bathed in intense radiation from Jupiter's powerful magnetic field, the strongest planetary magnetic field in the solar system. Jupiter's magnetosphere traps charged particles from the solar wind and accelerates them to tremendous energies. These trapped particles create radiation belts far more intense than Earth's Van Allen belts. Europa, the most scientifically compelling target in the outer solar system, due to its subsurface ocean and potential for harboring life, orbits within these intense radiation belts. Surface radiation doses at Europa reach several thousand millisieverts per day for unshielded surfaces. Even with heavy shielding, astronauts attempting surface operations at Europa would accumulate lethal doses within weeks. Ganymede and Callisto orbit farther from Jupiter in regions of lower radiation intensity. They represent somewhat more accessible targets from a radiation perspective, but transit times to the Jovian system exceed 3 years with current technology. Astronauts would accumulate career-ending doses simply traveling to Jupiter, regardless of what they did after arrival. The outer solar system offers no refuge from galactic cosmic rays and requires mission durations measured in decades. Saturn orbits at roughly 10 astronomical units from the Sun. Uranus orbits at 20 astronomical units. Neptune orbits at 30 astronomical units. Transit times to these destinations using any demonstrated propulsion technology span years to decades. No human could survive the transit to Saturn or beyond under any credible mission architecture. The cumulative radiation dose would be fatal long before arrival. A 10-year transit to Saturn would deliver roughly 4,000 to 9,000 millisieverts, depending on shielding effectiveness. This dose range spans from severe chronic exposure to acute lethal exposure. Astronauts would either die during the journey or arrive so radiation damaged that meaningful operations would be impossible. The outer solar system will remain the exclusive domain of robotic probes for the foreseeable future and likely forever. Machines do not accumulate biological damage. They can be hardened against radiation effects through redundant systems and radiation-tolerant components. They can operate for decades in environments that would kill humans within months. Interstellar travel represents the ultimate collision between human ambition and physical reality. The nearest star system, Proxima Centauri, lies 4.25 light-years away. This distance exceeds the distance to the Moon by a factor of roughly 100 million. It exceeds the distance to Mars at closest approach by a factor of roughly 7,000. The scale is almost incomprehensible, yet it represents the closest possible destination beyond our solar system. At 1% of the speed of light, an extraordinarily ambitious velocity target, far exceeding any demonstrated spacecraft capability. Transit time to Proxima Centauri would exceed 400 years. At 10% light speed, a velocity requiring propulsion technologies that remain entirely theoretical, the journey would take roughly 43 years. 43 years of deep space radiation exposure would deliver roughly 16,000 to 36,000 millisieverts, even with optimal shielding. The lower bound assumes best-case shielding performance throughout the journey. The upper bound reflects more realistic shielding limitations for a spacecraft that must also carry propulsion systems, life support, and crew provisions. Both bounds represent fatal doses. The lower bound would cause severe chronic radiation syndrome with progressive organ failure over the journey's duration. The upper bound would cause acute radiation syndrome within the first decade of travel, killing crew members long before the halfway point. No human crew could survive interstellar transit at any velocity achievable with foreseeable propulsion technology. The cumulative radiation dose would be fatal regardless of shielding, regardless of medical intervention, regardless of crew selection or mission design. The physics simply does not permit human survival across interstellar distances. Generational ships, often proposed in science fiction as a solution to the interstellar transit time problem, face the same constraint multiplied across multiple human lifetimes. The concept envisions a self-sustaining spacecraft carrying a population large enough to maintain genetic diversity across generations. Children would be born aboard the ship. They would grow up, reproduce, and die during the journey. Their descendants, generations hence, would arrive at the destination star system. Each generation would inherit the accumulated genetic damage of their parents and add their own lifetime exposure to the tally. Mutations in germ cells would be passed to offspring. Chromosomal abnormalities would compound across generations. The ship population would experience progressively shorter lifespans as accumulated genetic damage degraded biological function. Cancer rates would climb with each generation as tumor suppressor genes accumulated mutations faster than selection could remove them from the population. Cognitive function would decline as neural development occurred under constant radiation bombardment generation after generation. Fertility would decrease as reproductive tissues accumulated damage faster than repair mechanisms could compensate. By the time a generational ship reached even the nearest star, its population might be so damaged that establishing a viable colony would be impossible. The founders who departed Earth would condemn their descendants to lives of progressive biological deterioration aboard a vessel traveling toward a destination that healthy humans might never reach. Suspended animation or cryogenic preservation does not solve the radiation problem because radiation damage occurs regardless of metabolic state. A frozen astronaut accumulates dose just as a conscious astronaut does. The particles passing through frozen tissue cause the same ionizations they would cause in living tissue. DNA breaks form in frozen cells just as they form in metabolically active cells. The molecular damage is identical whether the cell is functioning or preserved. The only difference is that frozen cells cannot repair damage as it accumulates. Living cells continuously scan their DNA for breaks and attempt repairs. Frozen cells do nothing. They simply accumulate damage in a static state. When suspended astronauts are revived, they will face the biological consequences of every particle that traversed their tissues during preservation. Their DNA will contain breaks that accumulated over decades. Their cells will bear the marks of countless ionization events. The damage that living astronauts would have partially repaired as it occurred will instead be presented to repair mechanisms all at once upon revival. The repair systems will be overwhelmed. Cells will die or malfunction. Tissues will fail. The revived astronaut will face immediate health crises from damage that accumulated while they slept through decades of transit. Suspended animation does not pause the accumulation of radiation dose. It merely delays the reckoning until revival. This systematic examination of every proposed destination leads to an inescapable conclusion. There is no technological solution that eliminates cumulative radiation dose as the fundamental limit on human survival in deep space. Faster propulsion reduces transit time but cannot eliminate the radiation accumulated during travel. Even instantaneous transit, if such a thing were possible, would not help astronauts who must spend extended periods at destinations that lack Earth's protective magnetic field. Better shielding reduces dose rates, but cannot reduce them to safe levels for indefinite exposure. The physics of nuclear fragmentation ensures that some radiation penetrates any practical shield. The galactic cosmic ray flux ensures that this penetrating radiation accumulates continuously. Medical interventions mitigate damage, but cannot prevent the progressive accumulation that eventually overwhelms biological resilience. No drug prevents ionization. No therapy repairs all DNA breaks without error. No genetic modification eliminates radiation sensitivity without introducing other vulnerabilities. The universe itself imposes this limit through the physics of cosmic ray propagation and the biology of radiation damage to living tissue. Galactic cosmic rays are produced by supernova explosions and other violent cosmic events. They propagate throughout the galaxy, essentially forever, deflected by magnetic fields but never absorbed. They will continue filling interstellar and interplanetary space for as long as stars explode. Biological vulnerability to ionizing radiation results from the fundamental chemistry of life. DNA and proteins and lipid membranes are all subject to damage from ionizing radiation because they are all made of atoms that can be ionized. No conceivable alternative biochemistry eliminates this vulnerability. Any system capable of storing genetic information and carrying out metabolism will be vulnerable to radiation damage. These are not engineering problems awaiting clever solutions. They are fundamental constraints that operate regardless of human ingenuity or determination. The physics cannot be negotiated. The biology cannot be amended. What does this mean for human expansion into space? It means that Earth remains the only place in the known universe where humans can live healthy lives for normal human lifespans. Our planet's magnetic field and atmosphere provide protection that exists almost nowhere else. We evolved under this protection. Our biology assumes it. Without it, we sicken and die on timescales that preclude permanent habitation. Every departure from Earth's protection imposes a cost measured in accumulated radiation damage. That cost can be paid for short excursions. Astronauts can visit the Moon and return. They can transit to Mars and conduct surface operations. They can spend months or years in space, accepting the health consequences that follow. The cost becomes prohibitive for permanent settlement. Families cannot thrive across generations under radiation exposure rates hundreds of times higher than Earth's surface. Children cannot develop normally under constant particle bombardment. Populations cannot sustain themselves when cancer rates climb and life expectancy falls with each generation. The dream of humanity spreading across the cosmos, colonizing distant worlds, building civilizations among the stars, collides with the hard wall of cumulative radiation exposure. The dream is beautiful. It has motivated generations of scientists and engineers and explorers. It has inspired literature and art and philosophical reflection on humanity's place in the universe. But dreams do not change physics. Ambition does not repair DNA. Determination does not deflect cosmic rays. We can visit other worlds. We have done so and will do so again. We can send explorers who accept shortened lives and elevated disease risk for the privilege of standing on alien soil. We can learn about the universe through their sacrifice. We cannot send families who will thrive across generations. We cannot establish civilizations that grow and develop without Earth's protection. We cannot escape our home world in any permanent sense. This is not a failure of human courage or creativity. The engineers who design spacecraft and the astronauts who fly them demonstrate extraordinary courage and creativity with every mission. The scientists who study radiation effects and seek countermeasures work at the boundaries of human knowledge. It is a confrontation with physical reality that no amount of determination can change. The same cosmic processes that created the heavy elements in our bodies, stellar explosions that scatter iron and carbon and oxygen across space, also create the galactic cosmic rays that limit where those bodies can survive. We are made of stardust. We are also constrained by the violence that created that dust. There is a profound irony in humanity's cosmic situation. We evolved on a planet protected by magnetic fields and atmosphere from the very radiation that permeates the rest of the cosmos. This protection was not designed for us. It arose from the same physical processes that created Earth's iron core and volatile inventory, but it shaped our biology fundamentally. We are creatures of the shield. We developed intelligence on this sheltered island. We developed science and technology and the ambition to understand our place in the universe. We looked up at the stars for millennia and wondered what lay beyond our world. And in doing so, we discovered that the cosmos is hostile to life in ways that cannot be overcome by the methods that brought us so far. Engineering and technology and determination carried us to the moon. They cannot carry us to the stars. The same curiosity that drives us outward reveals the limits that confine us. Perhaps future discoveries will change this picture. Physics sometimes surprises us with phenomena that overturn established understanding. New particles might be discovered that somehow shield against cosmic rays. New forces might be found that deflect radiation without the mass and power requirements of magnetic systems. New technologies might emerge that address the radiation barrier in ways we cannot currently imagine. Science has been wrong before about what is possible. Things once considered impossible have become routine. Heavier than air flight seemed impossible until it was not. Nuclear energy seemed impossible until it was not. Space travel itself seemed impossible until rockets made it real. But science has also established limits that prove remarkably robust despite every attempt to circumvent them. The speed of light has remained absolute for over a century, despite persistent efforts to find loopholes. Every proposed mechanism for faster-than-light travel either violates known physics or requires exotic matter that probably does not exist. Thermodynamics has constrained engineering since the 19th century, regardless of creative approaches. Perpetual motion machines remain impossible. Heat cannot flow spontaneously from cold to hot. Entropy increases regardless of human wishes. The constraints stand. Some barriers are fundamental features of reality, rather than temporary obstacles awaiting technological solutions. They emerge from the basic nature of matter and energy, and the laws that govern their interactions. They will not yield to engineering because they are not engineering problems. Cumulative radiation dose may be such a barrier. The physics that creates cosmic rays arises from stellar evolution and supernova explosions, processes that will continue as long as stars exist. The biology that makes us vulnerable to radiation arises from the chemistry of life itself, from the way genetic information is stored and processed at the molecular level. These are not incidental features of our universe that future technology might address. They are fundamental aspects of how matter and energy and information behave. Changing them would require changing the basic nature of reality. For now, and perhaps forever, humans are children of Earth. We can venture out, briefly, at great cost, accepting the health consequences that our biology imposes. We can send our machines far further than we can go ourselves, exploring the solar system and beyond through robotic proxies that do not accumulate biological damage. We can learn about the universe from the safety of our planetary cradle. We can observe distant galaxies through telescopes. We can analyze samples returned by robotic spacecraft. We can study the cosmic microwave background and deduce conditions at the beginning of time. We can know the universe, even if we cannot inhabit it, but the universe beyond our magnetic shield will not welcome permanent human habitation. The ceaseless rain of cosmic particles does not care about human dreams. It does not respect human ambition or reward human determination. It simply exists, permeating all of deep space, accumulating in every human body that ventures beyond Earth's protection, determining through patient physics how long each of us can survive in the cosmic wilderness. The answer to how long a human could survive in deep space is measured not in possibilities, but in millisieverts. Each destination has its dose rate, determined by distance from the Sun and available shielding. Each mission has its duration, determined by propulsion capability and scientific objectives. Multiply them together, and you get the accumulated damage that sets the hard limit. For the Moon, a few years of continuous occupation before career limits are reached. For Mars transit, months that consume significant fractions of lifetime radiation allowances. For Mars settlement, a lifetime of elevated risk and premature decline that may not be sustainable across generations. For the outer solar system, nothing achievable with human crews. For the stars, nothing at all. This is not the ending humanity wanted for its cosmic story. We imagined ourselves spreading across the galaxy, building civilizations on distant worlds, eventually perhaps filling the universe with human presence and human consciousness. That vision has inspired and motivated for generations. But it may be the ending physics demands. The universe is vast and ancient and utterly indifferent to human preferences. Within it, we occupy one fragile world where conditions happen to permit our existence. Beyond that world lies beauty we can observe, knowledge we can gather, and radiation that will kill us if we stay too long. The hard limit is cumulative radiation dose. It determines mission durations and destination accessibility. It constrains settlement possibilities and generational futures. It defines the boundary between what humans can achieve in space and what remains forever beyond our reach. Everything else is commentary.